a senior editor at Nature Methods. Today we have two guests with us, Lorenz and Sally Temple. Dr. Temple is the president of the International Society for Stem Cell Research, or ISSCR, and she is the co-founder and scientific director of the Neural Stem Cell Institute based in Rensselaer, New York. Dr. Studer is the founder of the Center for Stem Cell Biology at Memorial Sloan Kettering Cancer Center in New York City, and he is also serves as the director there. Both are MacArthur Fellowship recipients, and both use stem cells to study neurobiology. And they're both keenly interested in translating their discoveries into potential therapies in the clinic ultimately. So it's my great pleasure to welcome you both. Thank you for coming.
Thank you.
I just wanted to start by asking about the ISSCR annual meeting. This is the biggest stem cell meeting of the year, and there's a lot of great science. So what is one or two trends or topics that really get you fired up in this year's meeting?
So I think one of the most exciting things that we're seeing now in the stem cell field is the progress into the clinic. And we're seeing that the attendees of the meeting are really interested in this. So we have threaded clinical advances throughout the meeting, but we've also started some new programs. For example, the day before, we have a satellite meeting which will showcase some of the advances in neurodegenerative disease, in immuno-oncology, and also ocular indications, because there's been a lot of advances in that area. So we're very excited. We've got fabulous people coming in, and the idea is that we're going to provide information that will be valuable for certainly physician scientists, also clinicians who might be thinking what is coming down the pike. And also for patients and patient advocates. So it should be accessible to a diverse audience, and so far we've got great attendance. We're anticipating a really good interactive discussion about these matters.
That's great. So what do you see are some of the most promising preclinical translational kind of areas that are moving towards the clinic, maybe especially in your area of neurobiology and neural disease?
Sure. I mean, there are a number of really exciting areas. In the area that I'm working, there is a lot of effort in the area of Parkinson's disease, where we now really just are at the verge of basically bringing the ideas to the clinic that took nearly 20 years now to develop. And there's so much excitement around that from the community, from the science, and even from the industry, actually, that now starts getting interest into seeing how that can be translated. And clearly, Parkinson's is following other fields as well. And Sally obviously has pioneered the application of stem cells in various eye disorders, degenerative disorders. And we have a lot of progress, also kind of at the verge of really taking off in the area of diabetes, for replacing basically beta cells for glucose control. So there is really this verge that we actually know now how to make the cells. I think that's really the main progress in the field. It's no longer just which protocol to use. We kind of know to do that. That's the next step. Can we really use those cells clinically? And I think the next 5 years is going to be very, very exciting, because we're going to see really how far can the promise take us in real life.
So what would you see are the biggest hurdles right now towards that last step of going into humans with disease and people who actually suffer from the disease? Do you see what are the biggest kind of bottlenecks?
I think it depends a bit on the disease. So there are disease-specific challenges where sometimes it's more difficult to know exactly what's needed, which cell is needed, which combination. But there are also shared challenges. And one shared challenge is obviously, how do you go from a lab experience, we worked in a lab for many years doing that right, to something that's a product. It's no longer just a lab generated, it's actually a product. So you need to know how do you specify, like when you get a drug that you buy in the pharmacy, there is all kind of labels on it, now I know exactly what's in it. With cells, you kind of have to get to that level of precision. They know exactly what's in the cell, you know exactly how it works in that context. And that's still a big step that we are often not trained to think about. So I think it's where you need the multidisciplinary input. It's a learning process from both sides, because cells are not drugs. And so we need to get our knowledge into that field to really know for the regulatory agencies how to actually regulate cells. On the other hand, we really need to learn again how to make product that's actually suitable for a patient has also a big responsibility, because clearly we have a big responsibility to do it right. And I think there's always a big concern if you don't do it right, that could be very difficult for the field as well. So we really need, we are at this verge of the cusp of moving forward, but I think we shouldn't shoot too quickly. Now we do need to do it in a very deliberate way, and again do it in a way where we can do it in hand with the regulatory agencies.
Yeah, I think one of the exciting things that we've been seeing in our area of retinal pigment epithelial replacement for age-related macular degeneration, what we're seeing is now several groups involved in this around the world. There's a group in London, in Israel, in Japan, several groups here in the US. Most are using pluripotent stem cells. We're using an adult stem cell that generates the retinal pigment epithelium. It's exciting because when you have a number of different approaches entering the clinic, now we can share what the results are. And that, I think, will lead to the greatest progress. And I think in your Parkinson's field also, you are sharing results with other.
Yeah, exactly. We've actually started several years back, where we're not even completed already. We had a consortium actually across 3 continents, Japan, in Europe, in the US, where the leading groups kind of discussed in a very kind of, it's kind of a competition, but also a friendly interaction, or really setting up standards of how to move the field forward in a responsible way. And we learned so much from each other, and it gives you much more confidence now if you see how the progress goes, what are the common challenges, and really also decide what is the right patient, what do we look for. I think that's, again, something we should learn together. We shouldn't just try to fight each other. It's really kind of a stem cell community. I think we really need to do that right.
Yeah.
So when you started your careers, did you ever conceive that you'd be at a point in your lifetime where stem cells could be used for therapy? I mean, it seemed a little science fiction not that long ago, but...
Yeah, well, I think I started earlier than Lorenz, and I will say that at the beginning it was extremely, people didn't really think about stem cells in the nervous system. And then it was very controversial when it was suggested that the adult central nervous system might have stem cells. So at the beginning it was really, really a new idea. And through that, we got to the point of defining the cells. We helped extract them from the embryonic mouse and demonstrate that they existed, and the whole thing coalesced. I think it became very exciting at that point. It wasn't at the beginning. I had no idea, but maybe...
I mean, for me, it was always the vision that we actually would like together, and the vision came from the fact at that time where we were working not yet with stem cells, but we worked with fetal cells. And so there was, for me, I never thought that in that stage fetal cells would be the way to go long term, but that we might be able with stem cell to get cells that can match the cells in a fetus being born, but doing them much more controlled, much more reproducible. And again, that was just an incredibly long journey. I mean, this idea was down by the time I was a medical student in 1996 now. And so it's now a good 20 years that we worked on this idea, and we went through many different stem cell types and conditions. And so it's really very kind of exciting to see that all this work now comes to a culmination where we actually can move forward and test it in vivo. But it was clearly a long journey, and I think, like many, discoveries that hopefully stick around longer now, it takes quite a long time.
It is at an exciting point, though, it seems. So, so I'm coming from a methods journal, so I have to ask, what along this long trajectory of your careers, were there 1 or 2 methods that really kind of most deeply affected the work in your lab and allowed you to move forward, or even for the field, especially in your specific areas?
Well, certainly for me, when we began looking at disease modeling, the IPS discovery was a revolution, and the publication of that. I think the method was so robust. We managed to do it in our lab second try. And that, I think, speaks to a lot, because sometimes methods are published and it can be hard to reproduce them in any lab. Whereas it was such a revolution to be able to make patient-specific, individual-specific lines for studying disease, like age-related macular degeneration, tauopathies.
For me, it was also on the cell side. It was basically both the ability to grow human embryonic stem cells and iPS cells, because that immediately made even the mouse work getting a different flavor, because we knew we would have a human cell eventually that could be translated. That was a big part, and then the other part was really just how to tame the cells, no? The protocols to do that, and that took us a long time. And we made at least some contribution ourselves that is, I think, was useful for the field, called dual SMAD inhibition. It's like a very simple protocol where you give the cells just 2 molecules, and they go from being pluripotent to neural. And that really helped us a lot, not only because it's very precise, but it allowed the process to be very timed. And so you can now, on top of this time, you can give all the signals that happen in the embryo in a very controlled way. And so it made things much more predictive. It was no longer kind of a random differentiation. It was a controlled differentiation. And that allows them to layer cell type and cell type on top. And at the end, I mean, again, our vision is that in a couple of years, you can make any cell type at any scale from pluripotent stem cells. And that's no longer a crazy idea. It's actually within reach, definitely. And I think these are all these methods that were really essential for all the various tissues of the body.
Mm-hmm.
And so for in vitro cell culture, for modeling disease in the dish, how far do you think we have come? Are cells grown this way very similar to cells in the body or in animal models? And where are the remaining gaps?
That's a key question. You know, from our studies in the retina, we find we can, from pluripotent cells, make really nice fetal-like cells. So the products are immature. And I think across the board, that's a major hurdle. We need to know how to make a mature product and an aged product very effectively.
Yeah, I agree. I think that's the main challenge. Well, we're very excited that you can now actually use new techniques. And again, the meeting is going to feature a lot of those techniques like single cell profiling, where you can actually, from an embryo, look exactly how the cells should look like, look at every gene expressed, and then match them with the stem cells. And they're actually pretty good. So we get pretty good, but they're always at that early stage. And as Sally said, I think that's going to be the next 5 or 10 years now, to figure out not only how to make the right cell type, but to make it of any age. That you make a fetal cell, a 5-year-old cell, a 20-year-old cell, a 100-year-old cell, and what's really different. And we don't really know truly, from a basic perspective, what makes them different at those different ages, but they clearly behave differently.
Yeah, and I guess methods like single-cell sequencing were also like letting you getting to answer questions about heterogeneity. I know that variability, especially early on, was a big issue in the field. But it seems like people are starting to get on top of some of these issues, using more advanced methods like that. So I guess for you, if you had a kind of a dream list of methods or goals, where would you put that in terms of technical advances or achieving? So beyond, there's maturation. What else comes to mind in terms of what you'd really like to see?
So I think robust ways to make more accurate models. And we will be showcasing a lot of organoid work, which is incredible and moving so fast. But even so, I envision being able to incorporate more cells, to put them in the correct spatial organization, because sometimes now organoids don't completely recapitulate the normal 3D architecture. Because that, I think it will have value for understanding development, organogenesis, of course for disease modeling that we care a lot about. And could these also be the source of tissues for a more complete tissue loss, a regenerative product in itself? So I see that as something that we really are striving towards.
Yeah, no, I agree. In particular, there are these fundamental limitations also with size, for example, that again, nobody has still figured out how to basically make a vascular system or something that would really allow a tissue to grow beyond what usually the diffusion barrier, where the organoids, they get to a certain size and then they just cannot grow much more. But the human brain is nearly 3 pounds of mass. No? So that's a fundamental limitation that cannot really easily be overcome right now. And so we need to have organoid technology together with tissue engineering and again other cell types to really see, can we at least get closer to that or make models in a systematic way. And there are actually some very promising efforts that are going to be showcased at the meeting, where these smaller units can be generated that look very, very similar in the liver or in the kidney to what actually a kidney would look like. And so I think that's a very exciting development that's going to be highlighted, but again, that can be taken probably to the next level over the coming years. Yeah.
Mm-hmm.
Have you personally had to master any new techniques recently? I mean, I imagine running centers and institutes, you probably don't have much time to actually step into the lab. Do you get any time in the lab now, or is it not really
So it's mostly, for me, looking down the microscope, I talk a lot about protocols, a lot about methods, because I do find that if I don't know things to that granularity, things can drift in the lab. So I feel it's very important to be involved. I also would say in experimental design, especially for newcomers and trainees in the lab, to really drill down to what is going to be done. And for the human stem cell work, because it takes so long, we have to plan things out. So we have calendars of what we're going to do when. So I think to that extent, it's we're involved. Yeah.
Yeah, and I mean, obviously, that's again another theme of the topic. There's so much new technology is coming along, and many of the technology we use in our lab, like CRISPR engineering, where you control not only genes, but you try to control epigenetics. And I'm not going to do it myself, but at least need to be knowing enough that we can help supervising people in the lab. And so definitely keeps you always fresh, this idea. But I think where I really can contribute is more like Sally. We have the eyes, we've seen it for 20 years, how the cells should look. And I think that really helps. Or even, again, we need to compare it to embryonic tissues. Again, sometimes still dissect the tissue myself. So there's a few things that I'm still doing in the lab, but it's getting rarer.
Yeah.
And both of you actually have spouses who are scientists, is that right? So what is that like? Has that helped your work, or?
Yeah, we were talking about it because there's quite a few stem cell scientists that are leading clinical efforts that have partners involved. And from my point of view, it was essential. My husband started as a research scientist, a biophysicist working in vision, and then went back to school, did an MD, is now a practicing retina surgeon, has been for many years. So he truly understands the science and the clinic, and entering the translational world, it was enormously beneficial to have him do this as a partner. And, when you wake up at 1 o'clock in the morning with an idea, you need to talk to somebody. He understands. So I don't think I could have done this alone without him. Yeah.
No, I mean, for us, it's a really very close partnership, both in private life now and in that research project. Particularly, we kind of met through the Parkinson's project, actually, also a long time ago, and we kind of saw it growing up. Isn't it like another child now? That kind of gets now to maturity soon, 20 years old, and hopefully becoming independent. And that's it's exactly that. And she's also a neurosurgeon now, and she might actually be the person implanting the cells into the patient. So it's even in the practical procedure, it's going to be a very close partnership. But on the same time, she's also a co-investigator of the project. So she knows both sides, the basic side and the translation side. And it's extremely helpful, I think, because you have always these discussions, and I think it also helps you then to communicate them better to funding agencies and to investors and to other people, because you really thought about that very deeply, not just from the scientist side, but from the clinical side. And I think it helps you get the broader perspective from the beginning, because ultimately you need a multidisciplinary team. So at least you have already one more partner on that road directly at home.
That's beautiful. So teamwork at home, teamwork in the lab. That's really nice. Well, I want to thank you both very much for coming by. It's nice to thank you for sharing your stories and for illuminating a little bit of what you find interesting in the field. Thank you very much.
Thanks so much.
Thank you.